Ocean productivity largely refers to the production of organic matter by phytoplankton. Phytoplankton are photoautotrophs, harvesting light to convert inorganic to organic carbon, and they supply this organic carbon to diverse heterotrophs, or organisms that obtain their energy solely from the respiration of organic matter. Open ocean heterotrophs include bacteria, as well as more complex single and multi-celled zooplankton or floating animals, nekton, swimming organisms including fish and marine mammals, and the benthos, or the seafloor community of organisms. Phytoplankton, also known as microalgae, are similar to terrestrial plants in that they contain chlorophyll and require sunlight in order to live and grow. Most phytoplankton are buoyant and float in the upper part of the ocean, where sunlight penetrates the water, also known as the euphotic zone. Phytoplankton also require inorganic nutrients such as nitrates, phosphates, and sulfur, which they convert into proteins, fats, and carbohydrates. Phytoplankton form the base of marine and freshwater food webs and are key players in the global carbon cycle. They account for more than half of global photosynthetic activity and the global oxygen production, despite amounting to only about 1% of the global plant biomass. The many nested cycles of carbon associated with ocean productivity are revealed by the following definitions as seen in this figure. The blue arrows representing net ecosystem production, the red nutrient recycling, and the green autotrophic respiration. The total amount of organic matter produced through photosynthesis in a given area or volume over a fixed period of time is termed gross primary productivity, or GPP. Thus, GPP refers to the total rate of organic carbon production by autotrophs, while respiration refers to energy-yielding oxidation of organic carbon back to carbon dioxide. Like all living organisms, phytoplankton and photosynthetic bacteria need to respire to maintain their life processes. So not all of the organic matter they produce is available for growth and reproduction. The total carbon fixed into organic biomass during photosynthesis, minus that part which is lost due to respiration, or R, in a given area or volume over a fixed period of time, is termed net primary productivity. In simpler terms, net primary production is gross primary production minus the autotroph's own rate of respiration. It is thus the rate at which the full metabolism of phytoplankton produces biomass. Measuring total inorganic carbon fixation by photosynthesis directly is challenging, so measuring or quantifying GPP can be difficult. One method of quantifying total primary productivity is to measure the production and consumption of oxygen by phytoplankton using the light and dark bottle method. Using this method, a sample of seawater is collected from a discrete depth in the water column and the initial concentration of dissolved oxygen is measured. The water sample is then divided into two bottles. One bottle is a clear or light bottle, and the other is a light-proof or dark bottle. The bottles are then exposed to ambient temperature and light conditions, usually by hanging them from a buoy at the original depth the discrete water sample was collected for a fixed period of time. After the incubation period, the bottles are removed from the water and the concentration of dissolved oxygen in each bottle is again measured. The basic premise that you need to understand for the light-dark bottle method is that in the presence of light, photoautotrophs undergo both photosynthesis and respiration. In the absence of light, or darkness, plants will only respire. The difference in the concentration of dissolved oxygen from the beginning to end of the incubation in the dark bottles can be used to calculate the oxygen consumed or the respiration rate. The difference in the concentration of dissolved oxygen from the beginning to end of the incubation in the light bottles can be used to calculate the net oxygen production rate, or the net primary production, NPP. Therefore, if we know respiration and NPP, we can solve for gross primary production, GPP. To measure dissolved oxygen concentrations in our samples, we will be using the Winkler titration method. To conduct the titration, a round bottle with glass stopper is filled with sample water by allowing the sample water to overflow the bottle for 2 to 3 minutes. Dissolved oxygen reagent powder pillows 1 and 2 are added to the bottle. The bottle is then inverted several times until the powders are dissolved. Flocculant, or floc, forms as a brownish-orange precipitate, indicating oxygen is present. The stopper is again removed and dissolved oxygen-3 reagent powder pillow is added. The bottle is then inverted several times until the powders are dissolved. The floc then dissolves and the sample turns yellow. 1 full measuring tube of sample is then added to a measurement bottle. Sodium thiosulfate solution is then added by drop. The drops are counted until the color changes from yellow to colorless, being sure to swirl between each drop. The number of drops added is equal to the test result of dissolved oxygen in your water sample in milligrams per liter. Due to COVID restrictions, we will be unable to conduct this lab in person. You will be given exosonde water column profile data taken from Santee Basin during days 1 and 2 of a hypothetical light-dark bottle experiment. You will also be given hypothetical Winkler titration data for corresponding light and dark bottles at discrete depths within the basin. This data will be used to interpret the findings of your experiment in a full lab report. Included with your data, a lab report template and grading rubric will be shared to guide you in your writing.